Today, our discussion will focus on the process of bacterial conjugation and its types. To start off, we define conjugation as the transfer of DNA by direct cell-to-cell contact. In this process, the important thing to take note of are the plasmids, which are small double-stranded independent DNA molecules. Some plasmids are classified as episomes, which can exist with or without being integrated into the chromosomes.
The process of bacterial conjugation is dependent in the conjugative plasmids. The best studied example of this is the F factor, which is genes responsible for attachment and transfer. Most information required is located at the TRA operon.
Now we shift our discussion to one type of conjugation, which is the F-positive, F-negative mating. In this type, gene transfer is unidirectional. F-positive or fertile cells are also called the donor cells. On the other hand, F-negative or non-fertile cells are considered as the recipients. This process in general is also non-reciprocal. To further explain this type, a sex pilus is needed in order for contact to be established between cells, and once this occurs, F-positive cells then prepare for transfer using a type 4 secretion.
Replication happens through rolling circle replication, where one strand of the DNA is cut and the free end is extended while the other end is displaced to form a tail. The single strand may become double-stranded through complementary strand synthesis.
Rolling cell replication is initiated by a protein complex termed relaxosome. The relaxosome cuts one strand of the F factor at a site called the ORIT, or the origin of transfer. An associated enzyme, relaxase, remains attached to the end. As the process of transfer ensues, double strands are produced. The donor retains parental DNA and its complement, and once complemented, recipient cells become F positive.
A second type of F-factor-mediated conjugation was then discovered. In this kind, the donor transfers chromosomal genes with great efficiency, unlike in the former where DNA transfer cannot take place. Also, in this type, recipient cells remain as F-negative. Since there is a high frequency of recombination, the second type is also referred to as the HFR conjugation, and its donor is called the HFR state.
In this process, a part of the plasmid is transferred first. Next, chromosomal genes are transferred. And lastly, the rest of the plasmid is transferred. Complete transfer occurs within approximately 100 minutes, but the conjugation bridge isn't capable of lasting that long. Therefore, the F factor is not transferred entirely, which explains why the F-negative cells do not become F-positive.
Another type of conjugation concerns the F plasmid being an episome. It leaves the chromosome and resumes as an autonomous factor. When an integrated plasmid incorrectly leaves the chromosome, it may take some genes, and this results in the formation of an abnormal F plasmid.
An F' cell retains its genes and behaves as an F-positive cell, binding only with F-negative cells. The recipient thus becomes an F' cell and a partially diploid merozygote.
We have talked about the major types of bacterial conjugation, and now we will give minor points of information regarding its other examples. Less is actually known about conjugative transfer in gram-positive bacteria, and in other microorganisms, no sex pilus is formed, but the adhesion takes place through other mechanisms that can be observed using special plasmid-encoded proteins.
So we have reached the final point of our discussion. Thank you for listening. We hope our lecture was able to help you about the general concepts of bacterial conjugation.